Welcome, everyone, to the Science Circle, our Saturday presentation series. And I am your speaker, Stephen Gasior, Stephen Zootfly in Second Life. And I am presenting my annual genome engineering or genome editing year in review, where I collect up some of the most interesting, in some cases newsworthy, in all cases impact worthy, but I also delve a little bit into those more technical side stories I think will have more of a future impact. So I am a scientist at a company called KWS Group, and I will, and actually am a practitioner within this space. My structure and the usual topics, I like to repeat and do the same ones on a nice sort of grouping where we talk about the therapies and things that are getting to consumers. I'll talk a tiny bit about some background, and then we'll talk about target genes or modified organisms. Again, why, when we have the power to edit genomes, why do we pick certain ones? What are the interesting ones that people are picking? We'll talk about delivery because, again, you can't really do CRISPR unless you can get the components where you need them to be inside organisms, inside cells, specifically at the DNA. And like many aspects of medicine or therapies, it really is always one of the biggest challenges to get things delivered in the right places at the right doses. I'll also talk about new tools. So that's been a trend that's really kind of an impactful one. And I think this year we're going to have a special intersection, and I'll highlight how we're talking about protein engineering and deep genome scanning as ways that are now impacting the field more routinely. Finally, cautionary tales, as well as a couple interesting aspects of the legal situation with CRISPR. So just one opening statement. I am a researcher at an ag biotech company. They're a seed company with ag biotech components called KWS Group. And again, I'm not here in any way to represent the company's positions, and nothing that I say or discuss should be construed as actual investment advice, and certainly nothing related to what the company is or is not doing. One thing also, I decided to give a little bit of the template. So again, CRISPR, molecular biology, these are complex topics. I'm trying to give you in an hour some of the most interesting things that happened over the year, which could easily take all day if we really wanted to. And so each one of my slides is structured in a way that you can get the basic lesson from any part of it. Again, the top always has the citation of the paper or perhaps a news headline. The title of that paper usually gives you some takeaway information. And then within the body of the slide, I'll usually have one or two pictures that gives typically a quote from the paper, a description of the major findings, and sometimes a little bit of information about the figure specifically to itself. And then at the bottom, there's always the green, too long, didn't read. What is the takeaway lesson from this particular slide? And so again, this is not necessarily trying to help everyone understand every aspect of CRISPR, but to give us a survey of what's been happening that's been most exciting over the year. And it's been, I have to say, this year has been really above all other ones from before, just some really amazing stuff. So the first one is therapy advances. And what we have here is a paper discussing a customized base editing therapy in which a child was born with a metabolic deficiency, which is typically lethal. And what I'm showing on the left-hand side is a diagram of the strategy of what the gene looks like, how they're trying to do the correction. And on the right is from this one individual patient, a study of how in its bloodstream, when it's untreated, there's an accumulation of ammonia in the blood. Now, again, the accumulation of ammonia can lead to lots of neurological defects even before death. And so it's important to treat and maintain these patients right away. But after 2 doses, you actually see a dramatic reduction in the ammonia in this patient. And what's also important with the paper details is how patients that are untreated have to have a lot of interventions of blood cleaning, as well as reduced restricted diet, and that this was actually ultimately corrected as well. The child ultimately uh, was treated, successfully treated, to be independent from therapies from this CRISPR delivery. And if you think this is the description I'm going to give about baby TJ without any pictures, then you think ill of me. So just to give a little bit more of a picture of this and what was really entailed is baby TJ was born with an extremely rare combination of alleles that leads to a deficiency in an amino processing gene. No, it's not a mitochondrial enzyme. It just turns out these are extremely rare. It's a 1.3 million likelihood that you'll have two recessive alleles. So again, there's only a few thousand patients that could be around, specifically in the carbonyl phosphate synthetase, or CPS1 gene. Again, there are a lot of metabolic diseases that people have. And we actually routinely screen in the United States for something like 50 with a very tiny blood prick from almost all newborn babies in modern hospitals in the country right now. This one's lethal immediately, if within a few days, if you don't start the intervention. And the story behind this is that, the doctor, and this is in Philadelphia, noticed this and just knew enough of what was going on to contact interested parties that have for years been thinking CRISPR is an amazing technology that we can have customized treatments with very early interventions. And what you see on the right are a couple of these doctors with baby TJ after, I think, a few weeks after the dosing. And, the amount of effort that went into all this, where people volunteered really a lot of effort, time, and expertise, was all collected and brought together in order to make this happen in a span of about, I think, just a few months, about 7 months. And so this is really amazing, because this really does start to fulfill what is the very best unique case of what CRISPR can do. So remember that we've had genome modifying technology before, but in order to customize it to any given use case, it would take independent research and development, and also importantly, keep in mind, completely independent regulatory approval. That anytime you're to do some new gene and engineer it for some new disease, that's a years-long case trial, decades-long opportunity. But since we now know from several other diseases like sickle cell anemia, some other blood diseases, we know that CRISPR delivery is generally safe. And so what we have now, and I think I want to talk a little bit more about Fyodor Urnov's perspective that he's written about this, is that we can develop a regulatory framework where all of the components of delivery that are used for a particular delivery, again, the delivery could change, but that CRISPR itself and this RNA, these are all considered safe. And then for any given situation, you just need to go back and double check that the specific targeting you're doing and the specific ability to correct or change the genome in some way is the only thing that needs some degree of special regulatory oversight and permission at that time. So, what Fyodor is showing in this slide, this is a paper from 2021. Imagine having everything in place, having a framework where we can come in, we discover something, we develop a new guide for it, and then we basically do a very rapid, shortened regulatory approval. And so I think that's the big story of the year, that we have CRISPR enabling the medical community to do something very unique that has never been really thought of or done before in the history. Is there anybody here who saw the news about baby TJ? Because it really was was pretty widely disseminated in a lot of news media and whatnot. But cute little kid in a family that really was at odds. Oh wait, did I say? Oh, it could be KJ. Oh, did I mess it up? Yeah, maybe it's KJ. KJ. I used to have a friend named TJ. Okay, so that was interesting. The other big therapy advance that I wanted to talk about this year. Now, again, this is a mouse model, a mouse study, but again, the thing that's a little bit different than some of the other therapies and advances I've talked about in the past is that typically, like in the previous example, we think of CRISPR as a way to correct some genetic information to fix a disease, or to maybe attack the genetics of a virus, and that's a way to inactivate that particular pathology. What's interesting in this case is that when it comes to transplanting tissues from one individual to another, or what's called a xenotransplantation, where maybe we have pig organs or monkey organs that we transfer into human, you typically will get graft rejection. Now, you can overcome graft rejection in theory if you also include what are called regulatory T cells. And so in this research, they were showing that if you do some CRISPR editing to disrupt a couple genes, including the HLA, which is the histocompatibility locus. Again, this is the part of the cells that says this is me, this is not me. I'm going to attack not me. That if you do some modifications of that within regulatory T cells, then they don't get destroyed by the new host immune system. And those regulatory T cells can protect the engrafted tissue or organ. And so, again, this is a really interesting example of how CRISPR can be used to enable other aspects of metabolic therapy, or of therapies for, again, a wide range of pathologies. So really, and again, another really interesting example of its usage. But again, just to keep in mind, this so far has just been done in mice. And again, I think this does fit in the context too that people are using CRISPR editing within pigs, within monkeys, to see if there are ways that we can make organs more acceptable to humans as well. In addition to trying to use regulatory T cells as a co-therapy. And one group that I always like to highlight and summarize is from the Innovative Genomics Institute. This is the Doudna, University of Berkeley, the West Coast group that formed a company to try and advance a lot of CRISPR therapies. They do a roundup in series talking about this, and so they talk about the progress that we have in various clinical trials. They're highlighting this. So we've talked about this in the past. The top one are blood disorders. Cascovy is now out there, and something that anyone with sickle cell trait has an opportunity to get. So it's been approved. We have some CRISPR for really chronic or recurrent recalcitrant to treatment uterine infections. Cancer, again, I think within cancer, though, they're also including, how we use T cells, chimeric T cells as a way to attack cancer. And that would be my guess. Some other blood infections and sepsis. HIV, again, we have these ways of attacking the virus itself with CRISPR, or removing the ability of the virus to get inside cells. And, lupus and autoimmunity, cardiovascular diseases are on there too. And so one thing that's kind of interesting to keep in mind, is that, trying to fight off bacterial infections or lupus and autoimmunity, these aren't necessarily big markets. But the fact that CRISPR can give you some avenues to do it is that you can provide and develop a therapy that still can be profitable within that. Yeah, no, I don't think we're at the rats of NIM state as of yet. But I will talk about some brain stuff when we get to the delivery advances. So for this particular year, I've decided to really tone down the background and giving people some of the information. What I really want to focus on is what are we having and asking CRISPR to do? And like I mentioned, the basic structure of it is that you have a protein that can home into a location in the genome. This is enabled by a co-component, again, a second component known as RNA, and that the RNA is programmable to be what's called complementary to finding a DNA. And this is kind of, in a sense, it's kind of like using a picture, right? If you have a picture, you can recognize someone who looks like the picture, and that's how we're doing it within these cells. I think of it a little bit like Google Earth or Apple Maps, right? You give it an address, you give it some information, and it just goes right to what that place is. And then also combine that with the technology of an Amazon delivery drone, right? That in many cases, you can ask Cas9 to do lots of different things. Now, its intrinsic ability, what it does in its normal cell and location and organism is it cuts DNA, right? It's a way of trying to keep viruses or other stuff from invading the cell. Well, we can do lots of other things. We can have it just make a nick. Instead of a double-strand break, make a nick, and that can be useful. But we can also ask it to activate genes, turn off genes, make very specific types of mutations. You can copy in small sequences. You can chew up whole chromosome arms. You can insert large chunks of DNA. So again, once you develop the basic homing ability, you can ask it to do lots of different things. And one thing to also keep in mind is that when it comes time for those cases where it's breaking DNA, it doesn't do anything to repair the DNA. That is the cellular response saying, something's wrong here. We got to do something about it, and then they fix it in their own way. Let's see now. Now this is a research paper, and I'm using a little bit more here in the review area in the capabilities. But this year was one of the first times that someone more cleanly and clearly demonstrated taking different versions of these CRISPR-Cas proteins and having them do different things in the same cell at the same time. And so this graphic from the paper is just showing how they're developing technologies that at the same time you can ask one CRISPR to turn genes on, just very specific target genes on. You can use another CRISPR to target other genes and turn them off. And then you can have a third component, a third different component that's actually doing stuff with the genetic engineering, changing the DNA itself. And so I'm not going to highlight the specifics of what they were doing in this case, but they found that they could get this to work at a pretty robust percentage of the time in their target cells and have different things going on at the same time. And as we go through the rest of this talk, and I talk in particular in the new tools area, I'm showing 3 different things here, and we're getting at the point where we're talking about 12, a couple dozen different ways and technologies that are potentially available at the same time. And again, Max Chatwar asks in chat, are these genes, these target genes for this on the same chromosome? No, they're all over the place, right? They can be on any chromosome you want them to be, as long as you're programming your guides to recognize that particular target site. So that's interesting. Now, again, you still need to find the ones that work, because this is not 100% efficient, but it's a pretty amazing thing that we can do. So I think this is the year we might really start recognizing what's called, again, the term you're going to hear more often is multiplexing, which is again, not talking about your abdominal plexus or anything like a six-pack. This is multiplexing, doing multiple things at the same time. Okay, so there were some exciting things, in particular with some modified organisms and target genes. And this is one that I think we'll see how it comes out in terms of the intellectual property and how often it works across the board. But Tagline mentioned mitochondria, and I want to remind people that when we think about eukaryotic cells, and with CRISPR, we're typically talking about the chromosomes, the genes that are part of the main organism's genome. But we have a couple small components, subcomponents that are organelles, sometimes called plastids, that do specialized things within the cell. So mitochondria, those are the energy centers of the cell. And those are things that can help us take oxygen and convert it into abundant chemical energy. The other ones in plants, they have mitochondria. They also have chloroplasts, and chloroplasts are specialized organelles that can take sunlight and at the same time take carbon dioxide and use all that abundant energy and that carbon dioxide building block to make sugars, to actually make usable energy chemicals for what cells want to do. That then mitochondria go back and say, oh, I'll take that. And so probably what is I think it's still considered the most abundant enzyme, and really the foundational cornerstone enzyme of this whole process of allowing plants to do what they do, and thus allow organisms like mammals eat plants and do what we do, is something called rubisco. And rubisco is essentially this carboxylation machinery that takes the carbon dioxide and incorporates it into a 3-carbon molecule that then can be used for other types of stuff. So this is the most critically important enzyme in the world, and it turns out it's actually fairly inefficient. It doesn't do that with a high catalytic turnover. And so it's always been a dream to try and engineer better rubiscos. And what has been generally recognized for a long time is it's really hard to do. Once you start trying to make it work faster or overexpress it, it all kind of falls apart. It's a finely tuned machinery that you can't mess with too much. And so this work that is coming out of Japan is that they made some targeted mutations based on structural information and whatnot to target rubisco. And again, this is a mustard plant. This is Arabidopsis, a model organism. And they modified the rubisco, which is in the mitochondria, right? This is something where you can't use CRISPR. They used a different genome editing tool called TALEN. TALEN does not require this RNA cofactor in order to do what it does, but it's also harder to program. And what you'll see is that on that line 2 for the blue modification and also for the orange modification, the R312C and D397N, sorry, I looked at the wrong one, the M309I and the D397N. These are single amino acid changes enabled by TALEN, and you can see this much greater amount of body mass to each of the plants. And so what we have here is potentially something that can just boost it was shown for Arabidopsis to boost the amount of incorporation and the ability to more rapidly incorporate that into a bigger body mass for the plant. Again, this energy requirements, it's just got much more energy available in the cell because of that conversion process of carbon dioxide into chemical molecules. So what's also interesting about this is something that should be generally applicable to all sorts of different plants. That would include agricultural-based plants. And so this is something that I think we'll see how it comes out in terms of how often it works and how well it works and how easy it is to do. But we might see a pretty big burst in how much things can actually be in terms of when size matters, how much more robustly we can get more agricultural plants out of this process. So that's pretty exciting. And again, the more we can get more plants and more body mass per acre, then this is useful sustainability-wise, because then we need fewer farming acres in order to make the food we eat as much as possible. All right. This is another interesting one too. So I want to highlight a couple things that, plants in terms of agriculture have been worked on and genetically modified and engineered since the 1980s. And we've had other technologies that are not as precise, but have enabled some really pretty amazing advances in agriculture. But livestock has particularly been kind of left behind in that space. So I want to highlight a couple of these where one, sheep. So sheep are used for their wool, for the hair they grow, and that is again one of the most ancient fabrics that we've had for a long time. And but one of the tricky things is that when they've tried to get sheep to breed more in order to have larger flocks of sheep, anytime they've tried to breed more children per ewe, it also compromises their wool quality. So there's something about the genetics and about the changes they've tried to breed out of it. And so in this case, there's a known gene that makes the ewe ovulate at a more rapid rate. And again, I don't know the physiology of this particular target gene, but when they modified it with base editing, they showed that they can modify them where they can actually have, like shown in the graph, that hashed line is they're having more children per ewe, and it's not compromising the wool quality. And so this is a really good example of how uh, genome editing allows you to do something that your typical techniques of breeding were not achievable. Oh, so Mist asks an interesting question. Is it compromising the ewe's health overall? So again, these have been around for a couple of years, a couple of litters. There really doesn't seem to be any other aspect or compromise or harm that's going on with them. Again, that can take some generations and some more time to analyze and look at. But yeah, it's not like Dolly the sheep that, when they cloned Dolly, she ultimately died from a viral infection. And there just seemed to be a lot of health aspects to how that cloning worked. And so um, that does not seem to be the case here. Again, they're also targeting a gene that's well known for improving fecundity. So it's not necessarily, you wouldn't target something that also is compromising their health. So this is a gene target that's been around. It's just it didn't complement having good wool at the same time. But good question, because that's something to keep an eye on and look at over the years. And here's another interesting one. So this is, again, this could also fit in the legalities, but this is the first approval of a genome edited animal for human consumption. And so this is a big one. We've had, no, don't get me wrong, salmon has been approved before, but that wasn't done with genome editing. That was a slightly different way of doing it. But so salmon have been around, but this is the first example of a genome edited animal. And sorry, this is not even the US. This is in the UK. Sorry, this is both the US and UK. These were UK derived pigs. And so the key here is what they're trying to do was one of the things that we have with modern husbandry is that diseases become more prevalent because they're more close to each other. And viral or bacterial or yeast infections can really compromise very quickly, the ability to make money off of the sale of these. And just there's a lot of time and intervention, a lot of money and intervention. And so developing pigs that are resistant to those types of diseases becomes, again, a really valuable way to deal with that issue. And so these are resistant to porcine reproductive and respiratory syndrome. And so that is that's a pretty amazing step. I have mentioned in the past, and they have worked on the basic technology in chickens to make them resistant to influenza, but this is the first one that's actually made it through the approval process. All right. Any other news that you guys might have heard about with genome engineering and animals? Again, this is not related to livestock. A plant that grows wool. Well, I mean, we do have cotton. Cotton's an interesting target too. All right. So who have heard about Romulus and Remus, the quote unquote dire wolves that have been brought back from extinction? Yes. Okay, so this is work by a company called Colossal Biosciences. They're based in Texas, and they're doing something. And I'm just going to put this in the local chat too: de-extinction, right? And this is the idea that there are things that existed in the past We actually can understand their genomes. So Neanderthals, we have the Neanderthal genome, we have woolly mammoth genomes, we have all these things that we understand their DNA, but they don't exist. And so are there ways that we can do that? In this particular case, the company says they restored a once eradicated species through the science of de-extinction. And that is, they took regular wolves and genome edited them to be dire wolves. They were implanted into a wolf mother, and then they were born. And so that's kind of the exciting thing they were talking about. And again, let's say they did genome edit. Again, they edited a decent number of genes. I think somewhere in the range of 8 genes were edited. Then another few had that were edited in bulk, and then a few more were changed. But here's been the response of scientists, especially, and I have these quotes in here where Jacqueline Gill writes, "Colossal Biosciences did not revive dire wolves. To see this work being done with such a casual disregard, not only for the truth, but for life itself, is genuinely abhorrent to me." And critics have pointed out that, there are a lot of differences between wolves and dire wolves genetically. And again, even though you can have a very small percentage of things changing within an organism, the timing, the extent, the expression levels, these are all things that really programmatically give you very different organisms. And I think that that's a very good point to make, is that these are not dire wolves. But, I think this is one of those very interesting in-between cases. Now, again, my too long didn't read is that this could be the future of just very super expensive pets. I think was it Mist who said she wants a whole pack of them? How much are you willing to pay for that? That's I think one big question. And so I do wonder if, to some degree, some of these in between cases are a little bit more for news, because it's also not clear what direwolves would do within the ecosystem. So the one case they're making for mammoths is that you could reintroduce mammoths at scale enough in certain regions where they used to exist, and that can actually help with the ecology, the turnover of plants and animal matter to help restore ecosystems. And to me, it's not clear that that would be true for dire wolves as well. But it's interesting to see. All right. I do have one more to come back to later, but I accidentally put that out of order, so we'll come back to that after we talk about delivery advances. All right. So this is another example, and I've talked about some of these in the past too, where the brain is a really important organ target for a variety of diseases, as well as a variety of other types of treatments. But it's because it is a very specialized organ, there are lots of delivery challenges in that it has the blood-brain barrier. So what this paper showed, and this is kind of one of the better ones that I've seen to combine a few things, is that they're using viruses, and these are viruses that can be tailored and are very good at targeting and getting into the brain. I've talked about this in mice before. What this paper is doing is they have a little bit better technology so that to keep all the materials compact and also provide a brain-specific expression pattern to the genome editing tools within the brain to treat, in this case, prion disease. Again, the disease is relatively less important, but they were able to show that they can target a brain gene in a very specific fashion, where it's an all-in-one vector, like all the components are on that, because typically the original Cas9 really didn't fit on one of these vectors. And so this is taking advantage of, again, other variations of Cas9 and making them work for this purpose. And that's what they were showing here. Is they're showing the targeting strategy with the viral vector on the left, and then on the right, they're showing that they are able to target a gene within the brains, and in this case, giving you some improvement in the signal intensity of a marker. Tagline mentions, viral particles into the nasal vault. Um, Again, I I'm not. I've definitely done some myself, some sub or posterior to the eye orbital injections. The problem is, if you don't, if your package isn't specific to a particular cell type, you still have this thing where eventually you're going to get stuff distributed in all the places you don't want it to, right? That if you have something that can't cross the blood-brain barrier, then it stays in the brain. If you have something that does cross the blood-brain barrier, it eventually will leak out to the rest of the system. So, a lot of it really does have to do with being able to get into and be specific to a particular cell type, regardless of where or how. It's hard to make up for that by having a good injection site. Um, let's see. Next one. So here's another one. Again, we talk about, this is another brain delivery technology. And other than viruses, we have these things called like various types of lipid nanoparticles. And these are little encapsulating sphericals that go around the package you're trying to deliver. And you can basically change the composition of that spherical in lots of different ways. So this was a unique one in compared to other ones that I've seen. And again, in this case, they're targeting a gene that's known to be involved in Parkinson's. And what they did here is a little different from what I've seen in the past. Is they're using a sugar to actually do the targeting. And so in the past, most of the technologies I've seen have been very specific targeting peptides. So again, things made out of amino acids that look like proteins, but using sugar in this case ends up being a really good way to get through the blood-brain barrier. And so that was what I thought was kind of unique about this one. But again, highlighting that there are lots of different ways of helping things get past the blood-brain barrier. All right, and then this is the last one for delivery advances, and this is another example that where it's just this out of the box thinking for a different way of using CRISPR and using the components to really help with the therapy without necessarily it being about the disease itself. And so what this graphic is showing is that when you make these lipid nanoparticles, right, you can actually use different types of lipids and different types of ways of coating it that that can give it different places that it goes and gets into in the cell. But it's really hard to know this, right? How do you know that this particular composition of lipids and peptides and sugars will go to the lungs versus the stomach versus the brain? You don't necessarily know that. But what's interesting is that they're now using CRISPR as a payload and it's barcoded. And so again, the thing that's really critical here is they're using stuff we're familiar with, like in grocery stores. If you've been at the hospital, they have those little scanners. And so these barcodes basically allow them to understand and demonstrate which composition is going to which target tissue. And so what they found here is that they found lung specific as well as liver specific compositions that they could determine were the case because of the CRISPR-Cas editing. So Sumo asks, how big are nanoparticles? Again, I think these are kind of on the size scale of like smaller than bacteria. So I think there's something, they would be typically in the range of say 5 nanometers in diameter, or sorry, 5 micrometers in diameter. Offhand, I don't know. Offhand, I didn't really look at the size scale, but these are things that, I mean, one way to kind of think about the sizing of them is that they just are usually monolayers of lipids, of a phosphohead and a lipid tail. So they're extremely small. Well, again, they different things can happen. Sometimes cells recognize these as something to engulf, to basically surround and engulf in that way. In a lot of cases, you're trying to get receptors where then the membrane just kind of merges with the cell target membrane and just dumps the payload into the cytoplasm. So, okay, so D. Listen, it looks like she looked it up, that the diameter of these can be, I was pretty close. So 10 nanometers, up to the scale of 3,500 micrometers. So that's actually fairly big to be on that large scale size. But 10 nanometers is a pretty good size. 50 to 1,000 nanometers in diameter. Yeah, so 1,000 nanometers would be 1 micrometer. Yeah, and again, something you can put a live delivery payload within a syringe and get it inside of people, or an IV bag. So, and Cyril asked, how big are the ones that cross the blood-brain barrier? And again, I don't offhand know, but again, the barrier, the blood-brain barrier is not always so much about size, but whether you have the permeability. And so that'd be my contention, main point there. All right, but let's move on. So here's the targeted gene and modified organism I had a little bit out of space. Again, every year I try and highlight some kind of off the wall, off the beaten path organism, and just highlight the fact that the ability to have CRISPR be such a unique and powerful tool means that you can have it work with organisms that otherwise don't have a lot of money or development or just kind of this general effort and scalability of all the tools available. And so what it really is important in highlighting is the fact that any organism can be a model organism with CRISPR and finding the right way to deliver stuff. And so in this case, the giant freshwater prawn was something they were showing they could do proof of concept to genome edit. And again, they're just looking at, and as you see in the diagram, on the left-hand side, a wild type has this fairly dark pigmentation within the I think the early zygote. On the right-hand side, they're showing some diminution of that pigment because of the gene editing. So again, this is my oddball organism of the year, the giant freshwater prawn. All right. In terms of new tools, so again, I always find this exciting. Again, I'm a basic scientist. I'm always very interested in the new and exciting ways that people are finding to do things. And this one's pretty exciting. This was published in Science, and they came up with the acronym TIGER-TAS. And again, if you look at the title of this paper, it tells you the equivalent key thing, which is that they found a programmable by RNA, an RNA-guided DNA targeting system that's able to make breaks in DNA. And from the quote that I have here is that they use structural mining, sequence profiling, and large language model clustering techniques to identify something similar to the CRISPR-Cas9 systems we already have, right? So asking in a very specific computational way, can we find these signatures of this structure that does the thing that we want it to do out there? And that's what they successfully did. And what they found was a family of RNA-guided DNA targeting systems from bacteriophages, viruses, and parasitic bacteria, which they termed tandem interspaced guide RNA associated systems. And that's what's interesting. This is bimodal. There's a component that does the homing, and then there's another component that does the DNA breaking. And that's a little different. So if you look at the diagram on the left-hand side, you'll notice some similarities with CRISPR and some differences. And that is one, the similarity, they use RNA, it's binding to complementary sequences. But the big difference is that you actually have two separate RNAs that are looking at different strands of the target and are opening up the bubble in different ways. And so, this was published later in the year. And so, and they did show that it works in eukaryotic cells. One of the big challenges for these systems a lot of the times is that they're from bacteria. They work great in bacteria, but they take some time to work in the more complicated eukaryotes. And in this case, they found that it worked actually quite well in human cells. And so this is a really exciting development because it's another tool. It might end up being a highly competitive tool to the way CRISPR is working. But again, a lot of this also comes down to what is the patent protection? What is the IP? How much do they expect people to pay for it? And we'll really see what consequences this has and how generally useful it ends up being within the same spaces we've been talking about with CRISPR. But hopefully, at a minimum, we can think of it as a great tool to add to the multiplexing. All right, some other new tools. These next two are fairly similar. What I'm showing on the left-hand side is the various height of the green bars is showing this technical improvement of a compound. So if you look at the third one from the left, that's a very short green bar. They took that particular version of the protein and got it to work, as well as uh, the three bars on the right showed a dramatic increase in activity, as well as the second bar from left is one that they were trying to compare it to. Now, what's really interesting about why they picked what's called, well, ISCB is that once people discovered Cas9, they then a lot of groups asked, well, what was the precursor to Cas9? What was Cas9 before it was Cas9? And what they discovered was the ISCB is a part of a transposon system that finds sequences and breaks DNA for transposons to move around. And so people have shown that you can take these ancestral versions of Cas9 and basically enhance them enough to have them be as efficient as a new tool. And so that's what they're showing here. And again, this is, like I mentioned, this combination of diverse organisms, rational design and tools, but also this complex and genetic diversity we have from our databases to help us look for new tools, then use protein engineering to make them look better. And again, I didn't talk too much about this like I have in the past, but Cas9 was the first CRISPR system that people developed into a tool for this. But there's a whole other category of these known as Cas12, very similar in how they work, derived from a different transposon system. But they've actually diversified a whole lot more in prokaryotic systems. And so there's a Cas12a, that was the first one that was discovered. And now we're at, I think, Cas12n. But what we're showing in this paper, and again, the graphs on the left are showing generally increasing bars that are a highlight of greater and greater activity. But they took Cas12j, where the native ones weren't really working all that well for the tools in the lab, and they did enough engineering, adding other components like exonucleases to turn it into a more usable tool in eukaryotic cells. And so, again, this is helping us mine through what we have, use protein engineering as a way to give us more tools to work with, and also changing the IP landscape. Here's another interesting one. So again, this is a very hardcore protein re-engineering effort. And they took a completely different class of nucleases. Again, these are things that have been known for a long time, where they recognize a specific sequence of DNA and they make a double strand break. But they're not programmable, right? They in and of themselves are not programmable. Once they have a sequence that they recognize, they've evolved to recognize, that's what they cut. And so what this group was doing was basically demonstrating that you can reprogram these with a very hefty amount of protein engineering, modification, mutagenesis, and show that you can change the target of one of these into a different one. And again, this can be useful. Again, remember that one of the advantages in that mitochondrial targeting is that you can't do that with Cas9 because it's really hard to get the RNA into mitochondria as well. So this is one of these things where certainly a great application and tool for doing mitochondrial and chloroplast editing potentially. The other thing that's actually very interesting is that they found this particular one supports really good recombination. And recombination is a method of being able to insert genes. So a lot of times you talk about the double strand break making a mutation, and that's useful. But in this case, being able to deliver a fresh payload into the genome with recombination is also a really useful, powerful tool. So we'll see what comes out of these. All right, so probably one of the last terms or words you thought I would use in one of these talks is Legos. But here to me is an interesting paper that came out, and again, they specifically applied it to a CRISPR workflow where they use this Lego Mind, or what's it called? Oh, I meant to look this up. Mindscape, I think, Lego Mindscape, where you can basically use programming languages and build Legos and has moving parts that you can basically have machines. You can have robotic machinery that you build from Legos that's programmable. And what this group showed is that you can actually have various workflows that include DNA purification, protein purification, transformation modifications, all built around these Lego Mindstorm platforms that does this really pretty nicely. Now again, a lot of well-funded research institutes and universities, they can pay the millions of dollars to have companies like Hamilton and other places build really robust, specific for molecular biology workflows. Those are very complicated but powerful machinery. So this is an interesting space. And I think, my too long didn't read for this one, just to point out is, do not discourage any child or man-child's hobby because you never know what that'll come out, what can come out of that. I do have a couple cautionary tales, and I'll just highlight these quickly because these have been ones I've kind of talked about before. Is that, when you're making a break or modifying DNA in some way, your intent is to do something very specific. You have this idea in mind of what change you want to have happen. But a lot of times within the cell, it's much messier than that. Yeah, no, and I should say I use the term man-child because I like that one. But definitely girls, women should play with Legos as well. It's not a gender-specific type of thing. So in this case, this was a paper doing a little bit more of a specific. No, I don't say. I'm just saying I wanted to use the term man-child, which was more specific to Legos, but that anybody who enjoys Legos, it's that child coming out, whether you're a man or a woman, it's that inner child. And so in the cautionary tale here, this is one of the first groups to really done I think a more specific way of quantifying the different types of aberrant things that get into double-strand breaks and can actually impact the quality or even kill the cells that you have within the output of your genome editing experiments. And so I'm just showing a little bit here again that you have the target is on the top, and then the graph, the bars on the bottom are being funneled through the gray shapes to say, oh, within this targeting region, we're getting lots of different things that end up inserting into these places. And so the thing to keep in mind is that, in the agriculture and livestock space, we can have stuff go badly. And if there are changes we didn't want, we can discard those so they don't become the product. But when we think a lot about therapies for humans, where it's an injection into a body, these become very prominent, important things to consider in any given case. And so I think the point here is that, they're trying to just help add to this body of knowledge of how often and how dangerous these changes, these unintended consequences can be. And the other one is also, again, something related to work I've been researching and talking about in the aging field, but that anytime you go around making a double-strand break, you actually can change the methylation patterns, and methylation is really important for saying whether a gene should be on or off. And there can be consequences when that goes wrong, and we know this to be true in the aging field, that a lot of aging is actually potentially related to all the wrong genes being on and all the wrong genes being off. And so what's being highlighted here is that in both these experiments, where it's blue, that is a normal methylation pattern. And where you see these shifts to red, what they're showing is that at a point on the DNA where there's this break being made, you're getting these shifts to less methylation. So Susie G asks about and mentions phosphorylation. And of course, DNA is phosphorylated. Deoxyribonucleic acids, its backbone, its chain is phosphorylation. But the phosphate groups are what's broken when I talk about double strand break. And then the methyl groups, the methyls aren't on the phosphates or the phosphorylation. Those are actually on the nucleotides, but they're really more a matter of trying to pattern proteins being on or off the DNA to say whether a gene should be on or off. Okay. So again, we're still trying to work through, and I think this is one reason why we think we can do lots of pretty amazing things in therapy, but there's always this very cautious way of moving forward because we don't always know all the things that are happening. All right, so legalities are the last thing I'll mention. And again, before I leave, one thing I want to give everyone here, people should be able to access both the PDF for my files as well as the citations. These are Google Docs that anybody can just go download and access, a PDF and a text. I know everyone wants their own direwolf, or at least Miss does, but not yet. You have to talk to Colossal Biosciences. So legalities. This is a really important development in that the European Union has for much time said, hey, if we're modifying genomes, there's a lot of stuff we have to say what we will or will not allow. And so this year is two things happen. The UK has now implemented new policies that genome editing and certain types of genetic changes that you make are not regulated. They're considered, a modified crop, but they're not considered a GMO. And Europe has finalized the ruling that they will be treating them the same way. Now, the implementation will take some time in Europe, but at least this was the year where they said, we are going to have a category of genetically modified plants that we're not going to regulate like a GMO. And so again, this really will be opening up, I think, the opportunities in Europe and American-based agricultural companies to implement these. And then another one that I just want to remind people I think is really interesting is that India has unveiled the world's first genome-edited rice. And of course, rice is a really critical part of their agricultural body. And this is a reminder that other countries are getting into the game too. But not only are they regulating them in a way that they can be put and planted in the ground and consumed by consumers, but they're also the ones making it, right? So this is not a technology that is only focused on the United States, Europe, Japan, and China, right? These are technologies where India, whose overall research installed base is much smaller. And yes, and rightly pointed out is that India has a population of about 1 billion people, and so they really represent a little bit over 1/10 of the world's population, and so they are a big player population-wise. All right, so in summary, 2025 was really, I think, our most amazing year yet. This demonstration with personalized medicine, the true leveraging of the power of the programmability of the tools to do a therapy for like a very rare disease. Again, people are not going to put money into the disease prevalence of 1 in 1.3 million, right? So how do you treat these people? And this is a way to say, we'll put money into the platform and then develop the specifics on a patient by patient basis. The protein engineering, I really want to highlight that when you look at finding new tools, we think about taking stuff that's a little bit under effect, just not quite ready for prime time, but turn them into an effective tool. Protein engineering and deep genomic knowledge are things that can help us create these new tools. And I think, especially with the lessening of regulations and more stuff coming out in plants and also with animals, we're really discovering this history of safe use. And we have lessened fears of scary science gone awry. That's giving us some more regulatory breathing room for these to come out. And I think I just want to also point out CRISPR meets Legos. So I think that's pretty exciting. So that's the end of my talk. I thank you for your attention. Glad you were able to to be here today. And now I'll just take any questions.
You mentioned a PDF. Could we get a URL, or how are you distributing that?
Oh, I just put that in the local chat, so I'll pop them in again. These are Google Drive links, and you can download the PDF and the references from those links.